Hello, everyone. Thank you so much for joining for this first online event of Fun Day Sunday by India Bioscience. And today we have Mohit Kumar Joli from ISC Bangalore as our guest. But before introducing him, I would like to talk a little bit about Fun Day Sunday, what it is, what is this initiative, and why we're doing this. So we have been organizing a lot of webinars. We do publish a lot of articles, and sometime in 2021, we thought that how to make science or research a little bit more fun and engaging. So so that we can break it into little pieces and take it, near the general public. So we started this Fun Day Sunday initiative where we requested for entries which could be a poem or could be illustration or could be videos or audios, which could be anything that delivers some message and has some content of science or biological sciences, which is appealing in some sense. Either it could be visually appealing or it could be it could be a good music. I'm still waiting for to receive some good music on science, but so far we have received science illustrations, science photographs, science poems, and this Fun Day Sunday initiative started in September 2021. Initially, we thought of making 10 posts. We post Fun Day Sunday every Sunday on Twitter, LinkedIn, Facebook, and Instagram. Even though we started aiming for 10 posts, ultimately we ended up posting for 6 months continuously every Sunday without a break. So it was 24 entries in total, starting from September 2021 to March 2022. So now that season 1 of Fun Day Sunday is over, we are heading to season 2 of Fun Day Sunday. And we are starting with this talk by Mohit on cancer cells and how he relate this to Bollywood memes. So I would just like to walk you through some entries of Fun Day Sunday entries from post from season 1. So the first post of Fun Day Sunday was by Rafiq Mabur. He submitted human neuron, coronavirus, and few other posts. Then we received this beautiful portrait of Janaki Ammal by Kanika Khanna. And you can see we have a poem in Hindi, which is like, usually we publish in English, but this was exception. So by Sodavi Sonam, and also some other illustrations, Giants by Ipsa Jain. And you can see these are some Twitter posts, which are many of them, there are 24 entries, so I could not put all of them. So this is not it. There are many others as well. So if you'd like, you can visit our Fun Day Sunday page on India Bioscience Research page. We'll send you the link at the end of this talk. And since season 2 is open now, please do submit your entry for season 2 of Fun Day Sunday. And we'd love to post your entry as well. Now, coming to today's talk. Today's talk is by Mohit Kumar Joli from ISC Bangalore, who is Assistant Professor at the Center for Biosystem Science and Engineering, PSSE, at ISC Bangalore. Mohit is very much enthusiastic about SciComm and also memes, how to relate memes to science. Here he will talk about his research on cancer cells, and we'll get to see how he can use memes to science. So Mohit, floor is yours. Okay, I'll stop sharing.
Thank you very much, Suchi. Just share my screen.
Just also quick housekeeping instruction I forgot to add. Please add your questions in the chat box. And at the end of the talk, we'll request you to turn on your video and audio, and you can ask your questions by yourself. Thank you.
Is my screen visible, Suchi?
Yes, Mohit.
Okay, great. Thanks so much, Suchi, for that kind introduction.
Mohit.
Yeah.
Maybe you can minimize my screen because I think it's also.
Right. Just 1 second. Just a second. whenever I try to minimize it, just does a mix.
Maybe I'll turn on my.
I'm not seeing an option to minimize.
Yeah, maybe I'll turn on my video. Yeah.
Let's see if that.
Sorry, turn off my video. Okay.
Okay, you can see my screen now, right?
Yes, Mohit. Yeah.
Okay, great. Thanks so much, Suchi. Thanks so much, India Bioscience team, for giving this opportunity to present some of our work in a rather unconventional manner. As Suchi mentioned, that PsyCom has been something that I'm very passionate about, and today it gives me an opportunity where my words collide in terms of what I do in research and what I try to do in terms of PsyCom more recently via means. Here's an opportunity to see if there can be a reasonable intersection of those and an attempt to communicate some of our work through some means to a interdisciplinary audience that has been gathered here. So thank you so much for joining on Sunday, and I hope that this ends up being a fun day as well for you. Okay, so before I go into details of research,
Mohit.
Yes, Suchi?
I don't see your screen somehow.
Is
it visible to you?
Yes, it is visible to me.
Okay, fine, yeah.
Okay, can you see Zilay on the disclaimer slide?
Yes, I can.
Okay, I'm not able to minimize the Zoom window. Do you see it as well?
Can you try swiping over that Zoom window? That might get rid of it. At least on Google Meet, it does swiping.
Okay, does it help?
Yeah, that's on now.
Okay,
Yeah,
great. Thank you. All right. Yeah, so before I go into details, just a few disclaimers. First of all, any resemblance to anyone living or dead is purely coincidental and is not intended to hurt anyone's feelings. So this is a pure science communication attempt. And second, and more important, I completely understand the human value of this deadly disease that pervades the world. So please treat whatever I'm talking about today purely as an ingenious science communication exercise. And this is in no way meant to belittle this deadly disease that many of us know have suffered through and various different lives that it has touched. Okay, so what our research focuses on is the process, is trying to understand and eventually control the process of metastasis, which is how cancer cells move from one organ to another. And that remains the cause of 90% of all cancer-related deaths even today. We do not have a clinical therapy that can target metastasis or prevent cancer cells from moving from one organ to another. And what you see in the diagram here is just certain examples of where cancer cells beginning in different organs can metastasize to. So prostate cancer cells often go to the bone, breast cancer cells often go to brain, lung, liver, and bone. And how these cancer cells move through our body is largely through this fantastic set of freeways in our body, that is blood vessels. So imagine cancer cells in a given organ. They would get onto this freeway, travel all over the body, take different exits at different organs, and attempt to grow tumors at those organs as well. Now, metastasis, thankfully, has a very high attrition rate. More than 99.9% of cancer cells that leave the primary tumor or the home organ are not able to form metastasis. And despite that fact, you see that metastasis claims 90% of all cancer-related deaths. So it's still a major clinical challenge. So not all cancer cells are inclined or always ready to metastasize. There are mechanisms in place that prevent cancer cells from leaving the primary tumor. These mechanisms are both acted upon through other cancer cells as well as the normal cells in the body or immune cells, which are trying to prevent this catastrophic step. But a few cancer cells are able to leave the primary tumor, and then they go on to this journey of metastasis, which actually ends up being a highly treacherous and difficult one for them. And the reasons why this journey is extremely challenging and therefore it has such high attrition or failure rates is because the environment around cancer cells is constantly changing, right? So the amount of oxygen they have available is changing, the amount of nutrients they have available is changing. They are faced constantly by the immune system, which is trying to kill these cancer cells. And even if they're able to complete the journey through circulation and reach a different organ, that different organ is a very different environment, right? So bone is a very different biophysical and biochemical environment as compared to prostate, for instance. So that is one of the major reasons why metastasis is such a high attrition journey. And you can think of cancer cells which are going through this journey as being highly facing very high selection pressures and eventually dying in this journey itself. Okay, so then the question becomes that how do that very small percentage, the minuscule percentage, survive? What strategies do they use to survive, and can we understand that better and eventually control metastasis? So one of the themes that has been recurring in the literature is that the cancer cells which are able to survive are basically able to adapt. They are able to change their properties in a rather quick manner and also in a reversible manner. Right? And that is leading to them being alive throughout the journey and eventually being able to grow a completely new tumor at a different organ. And that's why you might have heard that cancer patients often die of multiple organ failures because these cells are able to metastasize and form tumors at multiple organs. So the questions that we are interested in addressing as a research group is that how do cancer cells switch their states? And the technical term for that is phenotype. And how can we prevent this cell state switching from happening, and how can we prevent cancer cells from adapting in such an efficient manner so that we can eventually restrict metastasis? Now, one can think of two different ways in which this adaptation happens. So one is genetic in nature, which is that every time a cell divides, there are some errors in copying of DNA. And those errors then lead to some mutations or change in DNA sequence corresponding to specific genes, which then leads to changes in the function. Right? So this is a very slow process. You need multiple cell divisions. Typically these cell divisions happen once every 24 hours. And the typical time for which cancer cells are present in circulation is only about a week. So a cell has maybe 3 or 5 different occasions to be able to do that. And as we all know that DNA copying in cell division is a process which has extremely small rates of error of the order of 10 to the power -5, -6. So therefore, only 2 or 3 cell divisions are not really sufficient to be able to give rise to some very specific mutations which might be helpful. So again, the situation is similar to in this particular movie, Karan Arjun, when this character is waiting for her sons to be reborn and come again to the same place 30 years later and avenge whatever wrongs were done to her. Right? So it's a long wait, and that might happen in Bollywood movies, but unfortunately, that is not a very efficient way that happens in cancer. So the other mechanism one can think about is non-genetic, which means that you do not necessarily depend on DNA changes. But you can actually change your metabolism, or you can change the expression of various other genes through different mechanisms, and you can change the phenotype. So you don't have to wait for cell division, and this can happen in seconds or in minutes. And these changes are reversible, and these changes can also be stochastic in nature. So once a change has been made, you can always come back to it, and it is possible that that change is passed on to the next generation. So it doesn't preclude what the ability of genetic changes are there as well. Now, so clearly, more recently, people have been realizing that it's the non-genetic changes or phenotypic changes that are playing a more important role in metastasis than the genetic one. Now, one of the examples of that is this phenomena, which is extremely well studied, something called EMT, or epithelial to mesenchymal transition. Now, what is this phenomena? if you think about the most solid tumor, most common solid tumors, such as breast cancer, prostate cancer, liver cancer, lung cancer, etc., those are all epithelial organs, which means that the cells do not have the ability to move, they do not have the ability to invade, they're actually stuck in a very specific 3D geometry to perform certain specific functions. Now, when they lose these epithelial characteristics and they gain the characteristics of other type of cells in our bodies, such as red blood cells, which are mesenchymal in nature, they migrate, they invade, but they do not adhere to their neighbors. Right? So this transition where some of the epithelial cells take on some of the properties of the mesenchymal cells is what is called as epithelial to mesenchymal transition, or EMT in short. And this is important in enabling cancer cells to leave their home and begin the journey of metastasis. Now, once they have reached the other organ, after surviving through the treacherous journey during circulation, they no longer need to migrate, they no longer need to invade, rather they need to settle down and form another tumor. And that is when they need to regain the properties that they had lost, and they undergo the exact opposite transition of what is called MET, mesenchymal to epithelial transition. As many of you may know, EMT and MET is not something very specific to cancer. This happens multiple times during development of an organism, during wound healing, during injury repair, etc. Okay. Now, if one looks at what was known about the role of EMT and MET in the field of cancer, these different schematics or cartoon representations taken from multiple reviews that have been written in the field by experts for almost a decade. So what you clearly see here is that EMT is thought of as a very binary process. Cells are either epithelial or mesenchymal. And you also see that it has been thought about that EMT is reversible as well, and which might not necessarily be true. So there were a few reports which did talk about this metastable state somewhere in between. But again, when it comes to what is the final functional phenotype, it was always referred to as either epithelial or mesenchymal. So this is a question that we started to ask: is EMT a binary process? Right? In other words, kitne phenotype the? And the answer is Sardar do. Now, if you look carefully at what set of observations in the literature sort of forced us to think of EMT in a binary manner, it was that we were basically taking readings only at 2 time points, right? So if you're taking readings at only 2 time points, you'll only be able to categorize 2 states and therefore call it binary by default, even if there are additional states that exist in between, right? So in other words, kitne time points? 2. And if you have only 2 time points, you can only say that it's a binary process. But as we very well know, and this is beautifully illustrated in this example of COVID cases, if you have, say, if you look at the green curve, which shows the number of cases in India, if you look at what happens in June and what happens in March, again, the number of cases are comparable. But that doesn't mean there is a straight line between them and the number of cases never went up or never went down, right? Unless you have data in between, it becomes extremely difficult to interpolate. And you can miss out on very important features, such as the existence of a major wave that came in and swept up many lives. Okay, so then we started talking to our experimental colleagues and said, look, if you want to understand dynamics better, we need to have much more time point data, much more resolved data in between, right? Until then, what we can do is, okay, let's see if a mathematical modeling exercise can help. And we were also not sure what exactly it will lead to, but we said, okay, it's worth a try. Now, what do mathematical models do is that they are capable of taking inputs from different variables and how those variables influence each other, and then predict what the different possible outcomes of a given system are. Right? So for instance, weather predictions that you all see on televisions are predictions from a mathematical model. And this mathematical, and we trust these predictions to a reasonable extent these days. But it has taken a long time to get us there. Initially, when climate scientists and the entire academic community invested in this effort, started to collect experimental data, they were also simultaneously building mathematical models in terms of how these different variables that follow, ideal gas law, Newton's laws of motion, etc., how they influence each other, and can we make reasonable predictions? And of course, the first predictions were perhaps not as accurate, but that led to collecting more data and improving the model constantly, and so on and so forth. So it's a very, very iterative process. But clearly, in this particular example and many other examples, mathematical models have, paved the way in terms of making accurate predictions. So the questions that we started asking was, can we now take an attempt similar to predict the dynamics of EMT, and therefore better understand how exactly metastasis happens? So what we did was we looked at EMT literature. This is how we started around 10 years ago. And what molecules drive epithelial behavior, what molecules drive mesenchymal behavior, and we put together a network in terms of how these different molecules influence each other. They either increase the rate of production or decrease the rate of production, which is shown by arrows or hammerheads, respectively, called activation or repression specifically. So what we know is that if the levels of these two molecules is high, the cell is mesenchymal, and if the levels of these two molecules is high, the cells are epithelial. And what you see here are different numbers which represent some sort of input-output relationship that can be gained from experimental data. Okay, so what you can do is take this information and convert this into a set of differential equations. I'll not go into the technical details today, but what did our model predict? So here in this graph, the x-axis is an EMT-inducing signal, and the y-axis is a measure of EMT-ness. The solid blue lines show the stable states, which means those are the phenotypes that cells can attain. And the dotted red lines show unstable states, so we don't have to worry about those. All right, so what does this graph tell us? First of all, it tells us that there is an epithelial state with very low levels of cell that we already know. It also tells us that there is a mesenchymal state with very high levels of cell. That's also something we know. But it also mentions that in between those two extreme states, there is a third state possible, which has Zeb levels more than epithelial and less than mesenchymal. Now, if you project them in terms of behaviors, this means that the adhesion is not completely lost and migration has not been completely gained, right? So this is a state which can allow for cells to move collectively rather than moving as single cells. So, but again, this is just a prediction from the model. We need experimental validation to move ahead so that anyone would take this seriously. So then we collaborated with a set of lung cancer biologists at MD Anderson Cancer Center. And what you see here is a lung cancer cell line where these cells have been marked for EEC-adherent environmentin, which are corresponding epithelial and mesenchymal markers with red and green respectively. So what you see is that most cells co-express red and green, and they continue to do that over multiple passages in the lab. So what this tells us is that the hybrid phenotype, as predicted from the model, to be a stable state, can actually be stably maintained in the lab over multiple different cell culture passages. So this was the validation that we provided, and then there have been many more studies in the past 6 years showing that yes, EMT is not a binary state, and there are many states that can exist in between, not just one state. And interestingly, these studies have also claimed, many of these, that the hybrid states not only exist, but in fact, they are the most aggressive ones. If you get rid of them, you can actually reduce metastasis significantly. There have been various reports across different cancers that have shown this now. So in the past 10 years, the picture of EMT has changed from a binary phenomena to one with multiple intermediate states, which are perhaps the fittest for metastasis as well, right? So the hybrid state, it still maintains some properties of the epithelial state, but it also gains some properties of mesenchymal. So it's sort of sitting somewhere in between. And it has been shown by us and various other colleagues in the field across different cancers that these hybrid cells can form more tumors. These hybrid cells can evade immune system, they can resist chemotherapy, they can resist hybrid therapy. And interestingly, it has been shown that these are the most plastic cells and these are the most adaptable ones. And so next we wanted to understand is what makes hybrid cells so adaptive, right? So of course, when cells are more adaptive, so let's say for instance, a chemotherapy, which reaches a cancer cell, or a targeted therapy reaches a cancer cell, is trying to kill the cancer cell through various different mechanisms. But if that cell is plastic, if that cell is adaptable, it can change its behavior. And it has been shown that the ones which are most adaptive are in fact hybrid cells. And what can happen in that case is that the drug is basically ineffective, right? The drug molecule is like chal hua hai mere saath. I was sent here to kill the cell, but this cell has now completely changed its behavior, so I'm not able to do anything. Okay. So, and this is also evident in the clinical data that has been collected in the past 10 years about hybrid phenotypes. So for instance, in this particular data, what you see is the percentage of such hybrid cells when you look at primary tumor biopsies taken from patients. And the x-axis shows the different types of breast cancer, and TN stands for triple negative, which is the most aggressive form of breast cancer. And that clearly has the highest number of such hybrid cells. This is a study from a pathology group where they actually did three-dimensional reconstructions of various different such slices that we were just talking about, taken from primary tumors. And what you see here is that cells are basically not moving as single cells. They are predominantly moving as clusters. So this, and those clusters co-express epithelial and mesenchymal markers. So clearly this challenges the entire dogma of EMT being a binary process and single cells being the most important ones to form metastasis. Now we clearly know across different cancers that when they leave together, they form much more tumors. So united they stand, divided they fall. There have been other reports more recently which have claimed that whether you look at overall survival, which means how long a patient lives, or disease-free survival, which means how long a patient does not have cancer again after he or she has been treated. If the number of hybrid cells in that patient is more than 2%, a very small number, just 2%, that drops down the time for which they will be alive, or the time for which they will not have cancer again, is drastically lower, which is what is shown in these two graphs, right? So you don't need to have hybrid cells in abundance. Just a very small percentage of them is able to do the job, and that can possibly still explain that even if 99.9% of the cells die, if there is 0.01% of hybrid cells, which are most plastic, most adaptable, they are sufficient to lead to metastasis and therefore perhaps much worse survival. So then we started to ask this question, that why are hybrid cells more plastic? What properties of the networks that control the ability of a cell to be epithelial, mesenchymal, or hybrid actually make them most plastic? And can we target that and reduce the fitness of cancer cell population? So before I go into that, I'd also like to mention that in the past 10 years, since we started the mathematical modeling of EMT, there have been various other mathematical models that have come out. Each of them uses a slightly different modeling method, slightly different biological network that they look at, etc. And they have somewhat different predictions. But a common prediction that exists throughout is that EMT is not binary, is that there are one or more than one hybrid states, and some of them also show that the hybrid states are in fact more plastic as compared to others. But no one goes into details of why are they more plastic. So what we have done here is that we have taken 5 of these networks, which I briefly talked about in the previous slide, and then looked at their properties. So I'll walk you through what properties we have looked at. So what we have done here is marked all epithelial molecules, which we know from literature correspond to an epithelial behavior, in red, all mesenchymal molecules in blue. So, and any activation is shown by green, any inhibition is shown by red. So what you see here, which is predominantly most clear in this 57 node, 113 edge network on the right bottom, is that a mesenchymal molecule always activates another mesenchymal molecule. So you always see green edges going among all the blue nodes. Similarly, if you look at any edge that goes from one blue to one red or one red to one blue, it's often red in nature, which means that an epithelial one inhibits a mesenchymal and a mesenchymal inhibits, an epithelium inhibits a mesenchymal and a mesenchymal inhibits an epithelium. Okay, so what this begins to tell us is that it is possible that there are two teams of players, one which is supporting an epithelial state, one which is supporting a mesenchymal state. So all molecules in one team support each other, and all molecules across two teams inhibit each other. So it's like a tug of war that is going on in this particular scenario. Okay, but that's just a visual, eyeballing the data. Let's look at it more carefully and see if this is indeed true. So what we then did is converted these networks into these matrices. What this matrix tells, let's look at the adjacency matrix first. So here you have every row is the source and every column is the target. So you have converted that graph into this matrix. So whenever there is a red box filled, which means that the row molecule is activating the column molecule. And whenever there is blue, it means that the row molecule is inhibiting the corresponding column molecule. Whenever there is white, which means that there is no direct edge that exists between them. So there are only 82 edges, and there are 22 multiplied by 22 combinations. So as you can see, this matrix is quite sparse. Now, this matrix only has direct edges, right? But one biological molecule can influence another biological molecule through indirect paths as well. So let's say node A can directly activate or inhibit node B, but also it can influence it through C. So node A activates C and node C inhibits B. So effectively, positive multiplied by negative, node A inhibits node B. Okay. So then what we did is that we multiplied this matrix multiple times to capture such indirect paths. And then we took an average of those paths. So what you see on the right in the influence matrix is not just the impact that node 1 has on node 2 through a direct interaction, but also through various possible indirect interactions. And we, of course, penalize for the length. The longer a path is, the less its ability to impact. Okay. So what you see here is a very clear pattern. All epithelial nodes shown in red squares, highlighted in red squares in column and the row, are activating every other epithelial molecule, shown by G22. Similarly, G11 means that all mesenchymal are activating all other mesenchymal. But if you look at G12 and G21, which means that every epithelial is inhibiting a mesenchymal and every mesenchymal is inhibiting an epithelial, right? So whatever we were just talking about in terms of eyeballing on the previous slide clearly shows up here. So it's basically two teams of players which are, which are supporting each other, and then across the teams they are inhibiting each other. Now, please note that there is a team that supports epithelial, another team that supports mesenchymal. There is no team yet that we have seen for hybrid. So then we wanted to ask, is this the reason why hybrid cells are more plastic? Because there's no one supporting it. So, there is a bunch of hybrid cells sitting in between. And if you pull them to being epithelial, they'll become epithelial. If you pull them to being mesenchymal, they become mesenchymal. There's no one to support them. So then we wanted to ask that, is this presence of teams specific to the EMT networks, or is this just an artifact? So what we did here is, just as you would shuffle a deck of cards, we took a biological network that I talked to you about and then shuffled edges among them. So now you have many, many other networks which are non-biological in nature, which are hypothetical in nature, but they have the same number of nodes, the same number of edges, but a different influence matrix and a different adjacency matrix. And then we asked, okay, what is the strongest possible team present in those networks? We took 1,000 such random networks and said, okay, if this is the strongest team that these networks have, is this in any way comparable to the team that we actually saw in biological network? And here is the answer. The answer is that it's not. If you look at the red line shows the biological network, and the histogram shows the distribution of team strength seen in random networks. So you can clearly see that the biological network has much higher team strength as compared to most of the other non-biological networks. So this is EMT networks telling their randomized counterparts, "Ki beta tumse ho nahi payega," right? If you're depriving me of metrology, I will deprive you of the function, right? So structure dictates function, kind of adage shows up here as well. Okay, so then we ask that, if there is a team, then if you start from an epithelial phenotype and you try to gently push it to mesenchymal or pull it out of an epithelial state, then there should be some resistance. And similarly, there should be resistance for mesenchymal, but there should be minimal resistance for hybrid because there is no hybrid team. And that is what we saw, as you can see in this diagram. The x-axis is the amount of perturbation, the amount of force in some sense that you're applying to pull it out by changing the number of nodes from positive to negative or negative to positive. And, whenever you begin to see that things are dark blue, which means that that state no longer exists, largely speaking. So you can see that boundary comes around 40% on the x-axis for red and blue, the epithelial and mesenchymal. But for green, the middle patch, that boundary is somewhere around 4 to 5 or 10%, right? So we quantified this further and we showed that no matter which network you look at, it is always easier to pull out hybrid as compared to pulling out epithelial or mesenchymal. So then we wanted to ask, okay, this is a property of the network, but how do we show that this is happening because of the presence of teams? So then we took one of the random networks that I was talking to you about and did the same experiment. So in a biological network shown on top, you see that initially there is some resistance. So the y-axis shows how far away you are from where you started, and the x-axis shows the amount of perturbation. So it's not a linear graph. Initially there is some resistance, then it sharply increases, and then it saturates again. So that resistance is coming into teams. But if you look at a random network which does not have such teams, you see more or less a linear behavior, right? So for every amount of force that you're applying, you're slowly moving away the system from where you started. There is no concept of resistance that sort of shows in there. Okay. So in summary, what we found was that there are teams which stabilize epithelial phenotypes, which stabilize mesenchymal phenotypes, but there's no team that supports hybrid phenotypes. And that is the reason why hybrids ones are actually more metastatic, are more plastic and perhaps more metastatic. We have looked at some clinical data as well. This is unpublished, where we do see the evidence that patients who have more metastasis do show weaker teams as compared to patients that do not have metastasis. So clearly, team strength is playing a role in the entire picture. Okay, so I'll just summarize this aspects that we just talked about, that metastasis is thankfully a highly inefficient process. Cancer cells that are able to metastasize are among the most adaptive, and hybrid cells clearly have been shown to be more adaptive and more aggressive. And one of the reasons we think they are more plastic, more adaptive, more aggressive is that there is no team that supports them. For epithelial and mesenchymal, there are these teams that can support them. Okay, so what do cancer cells teach us? Right? If you look through this journey of these cells, they are constantly changing. They are constantly adapting. They are not waiting for cell divisions to happen. They're not waiting for next generation will take care of this. They are themselves doing it in a very quick manner. Right? So clearly, they're keeping their options open and they're being in the moment. They're basically performing improv. So that's one lesson that they teach us. Then there is a lot of cooperativity, a lot of collective behavior, a lot of teamwork. We already talked about the existence of teams in terms of nodes within a cell, but there is other evidence that even cells of different type can cooperate. And again, cancer cells or tumors which show evidence of cooperation across cells do much worse. Those patients have much worse outcome as compared to cancer tumors where there is less evidence of such cooperation, right? So there is cooperation happening at all different levels of, and clearly the ones that leave as a cluster form much more metastasis, right? So they're basically telling us that, jhund bana ke chalo, and that's how you'll be able to survive. The other, this is the most important lesson, according to me, is that cancer cells are not worried about what you call them. You call them epithelial, you call them mesenchymal, they don't care. They are there to perform their function. They are there to metastasize. They are there to disrupt the organ architecture. Okay, so we place names on them. We place categories on them because of our limited understanding. Okay? And through the example of EMT, we have clearly seen that boxing them in two discrete bins was actually quite counterproductive for understanding things better. Right? So we need to be careful about this, this habit of categorizing them as much as possible. This categorization might be helpful, but only to a certain extent. We should sometimes place ourselves in the feet of cancer cells and think that, look, they are not really worried about what you call them. So therefore, let's not worry too much about the labels. Let's worry more about the properties. Okay. Similarly, metabolism has been thought of as initially as a binary axis, but now we know that's not the case. There are cells which can do both glycolysis as well as oxidative phosphorylation. It's not as black and white as we often presume things to be. The question that becomes is that, going back to how cancer cells are able to metastasize, is that they should be able to change multiple properties simultaneously. So when they're beginning to move, they should be able to also change their metabolism and so on. So how are these properties coupled to each other? And can one drive the another? Can second drive the one? These are all interconnections that we have very little understanding of. So I'll give one example that we recently started looking into in this particular case, which is the connection between EMT and drug resistance. So this is the example from breast cancer, where there's a specific drug, tamoxifen, given to this subset of breast cancer patients. And there was some associative evidence clinically showing that patients who do not respond to this drug have more mesenchymal behavior. But we wanted to understand this, what is causing what? And again, can mathematical models help? So we put together a similar network based on literature. And when you simulate the dynamics of this network, what this tells you is that EMT can actually drive resistance to this drug, right? And then we did some transcriptomic data analysis on published data and showed that whatever our model predicted was in fact captured in there. So what this means is that when cancer cells are leaving the primary tumor, they are already carrying together the property to be able to resist the drugs, and vice versa, that resistance can drive EMT. So if you continue to treat patients constantly with very high levels of this drug, you're putting very high selection pressures, which is leading for some cells to become resistant and at the same time metastatic, right? So drug is being counterproductive here. Drug is in fact accelerating the process of metastasis, which also has been reported in many cases. So what our model predicted is that one should give these two drugs together, tamoxifen as well as inhibitors of EMT or inducers of MET, and these predictions are currently undergoing experimental validations. I'll just summarize and end with this, this main point, which I started with, is that we need to think about cancer as a complex, dynamic, and adaptive system. We cannot rely on a binary understanding. We cannot rely on a static understanding of this. And there have been unfortunate instances in the clinic where where we have thought of cancer as a static system, and that has led to counterproductive results. So for instance, in the early 2000s, when the idea was that you block cancer cells, you block the growth of blood vessels around the cancer cells, and you can take care of cancer growth. This drug was given, clinical trials were started, and the clinical trials had to be stopped because patients receiving this drug started dying much faster. 10 years later, we realized. Yes,
Mohit, you have 3 more minutes.
I'll be done in 2 minutes. Yeah.
Okay, sure. Thank you.
So then they realized that this was happening because when you are blocking them from growing blood vessels, you are actually increasing their metastasis. So cancer cells are clearly very adaptive in dynamics, and we should look at that accordingly. And that's where I think mathematical models can play an important role, because you can go back and forth with experimental data. With that, I would like to thank the fantastic set of people I've had the fortune to work with, who come across different disciplines and enrich our dynamic understanding. And of course, the excellent set of team members here at ISC, BSSC, who continue to push our understanding of these processes better. Thank you so much, and I'll be very happy to take any questions.
Thanks, Mohit. Could you please stop sharing your screen? Yeah. Thanks a lot for this engaging talk. And I guess my take-home message today will be keep your options open like cancer cells and be adaptive, right? Adapt. Okay. So do we have any questions for Mohit? Shantala, would you like to turn on your video and ask this question yourself? Okay. Pallavi Mitra is asking, what was teams that was talked about? Is it a team of molecules or networks or something else?
Right. So it's a team of nodes in a network that are acting together. So these nodes, even if you delete one of these nodes, there are other nodes which can take up the same job and act as a backup in some sense. And they're overall resisting the ability of a cell to change its phenotype. So this can prevent epithelial cells and mesenchymal cells from being more plastic, but because there is no team for hybrid, the hybrid cells get pulled off in either direction easily.
If you would like to ask questions to Mohit directly, you can raise your hand and you can turn on your video and ask questions. Somilia Mishra, would you like to ask the question? Okay. Somilia Mishra?
Yes, sure. Am I audible?
Yes.
Yes. Yeah, it was really a very wonderful talk, sir. And my small question is how can these mathematical models can be used in early detection of cancers?
Okay, so that's a great point, Somilia. While my work is not directly focused on early detection, what I've seen in the literature is that people use changes in histology of an organ as well, and try to identify, what histological changes happen at different stages of cancer progression, and through more quantitative analysis of that, can one predict at what stage a given patient might be. So there are some analysis towards that. And more recently, with the single cell RNA-seq technology coming in a big way, there are computer scientists who are trying to predict when exactly a pre-malignant tissue becomes malignant based on changes in gene expression.
Thank you, sir. Thank you, sir. I got my answer.
Thank you.
There's a question from Shantala, which is, does this model play a role in normal development as well?
Great point, Shantala. Yes, it does. We have not customized our model to look at EMT and MET in development yet, but we are working with experimental developmental biology groups now to better understand how is the EMT-MET dynamics in development different than that in cancer.
Okay. We have some questions from the registration forms as well. People submitted some questions, so I'll ask something from there, which is a light-hearted question. Why memes? It is from Manikant Roy. Like, why you're using memes to explain science?
That's that's a great question. I think memes can be a good way of message reaching to a larger audience than just a specific set of experts thinking about the same topic day in and day out, just as you. And of course, humor has an important role to play in public communication. So those are the two reasons.
Okay, yeah. So there's one more question from Nandan R. Barade. Is there a possibility of vaccine for cancer disease?
Yes, so for certain cancers for which there have been important investigations where virus or oncoviruses or virus-induced cancers, for those particular diseases, there is increasing research and implementation in terms of cancer vaccines, but I don't think it's very generic throughout.
Okay. Vijay Jayaraman, got one question from Vijay Jayaraman. Vijay, would you like to turn on your video and ask or audio and ask the question? Okay, sure. You can turn on your audio also if you'd like. Sure. So I'll ask the question. Do you know of?
Hi, Mohit. Sorry, yeah.
Okay, yeah, please continue.
Yeah. So do you know of any network that can, with such binary teams, that can promote hybrid formation? And how will they look like in an adjacency matrix that you showed?
Right.
Are there any natural examples? Yeah.
Yeah, so we are looking at instances of 3 cell fates coming from the same progenitor now to identify how the adjacency matrix would be. So the example we have picked up so far is that of T cell differentiation, where there are multiple states coming from the naive T cell. So there we do see that in the adjacency matrix, just as you saw 2 teams, G11 and G22, we do see G11, G22, and G33. We are still at very initial stages of understanding how this network behaves differently than the network that has only 2 teams. But yes, there are examples that we can look into, and we have begun to look into.
Wonderful. Thanks, man.
Thanks, Jay.
We'll take one more questions from the registration forms, which is, what is the efficiency of nanotherapy in terms of cancer? It's from Abhishek Jha.
So that's a very good point. I think there is increasing evidence of nanoparticles being used for both for targeted delivery as well as for sort of more efficient packaging of the drug. So yes, again, that is, I'm not aware of where they are in terms of clinical trials and implementation into the clinic, but those 2 directions clearly have shown a lot of promise.
Okay. So Ajka Masi, he asked that so far, what is the recovery rate for this disease? And what are the modern techniques to get rid of this disease? And the most important, how can we keep ourselves safe?
So there are various lifestyle factors clearly which have been associated with different types of cancer incidents. So for instance, smoking causes lung cancer, and particularly one of the most aggressive forms of lung cancer called small cell lung cancer has been shown to be present exclusively in smokers. So there are lifestyle factors which one can try to modulate accordingly. In terms of recovery rates, again, the way these are clinically measured is something called 5-year survival rate, which means that out of 100 patients, if one looks at what percentage of those patients were alive 5 years after diagnosis. So this percentage is reasonably high for, say, most forms of breast cancer, which is, when I say reasonably high, I mean more than 90%. But this percentage is abysmally low for other cancers, such as pancreatic cancer, where this is less than 10%. So there are various reasons. Pancreatic cancer is harder to detect in earlier stages. We still do not have as well targeted therapies, etc. But yes, the cancer's aggressiveness has a very wide spectrum.
There are some science communication questions for you, Mohit. One is, what is the golden rule for science communication according to you? This is from Megha Nivaskar.
So I think the rule I try to follow is the same for any communication exercise, including science communication, is know your audience and know what they expect of you, and try to then sort of modulate your message accordingly in the amount of time that you're allotted.
Okay, one last question, which is about the motive of today's talk. And the question is, how can we make research enjoyable? And it is from Sentu Ruja.
I mean, research is enjoyable in itself for multiple reasons, for sure. But yes, I think the way many times it is communicated largely to peer community is in very academic and formal terms, and that can limit the enjoyability part of it at times. But there are various other science communication initiatives which are taken with different audience in mind, from school children to college-going students to even more senior adults in different forms, in theater, in music. There are competitions such as Dance Your PhD, and I'm sure through Funday Sunday also the message is reaching to many other people who would not have necessarily read a research paper otherwise. So yeah, all these options exist, and whichever one people feel most comfortable to use, I think they should use it as much as possible.
I'm sure research is enjoyable by many people without any reasons as well. They do research because they enjoy research, right? But yes, we can use some tactics to make it more enjoyable. We will take one last question, which is from Samilia Mishra. Samilia, if you'd like to ask a question, you can turn on your audio and
Hello, sir. One last question related to mathematical models only. Sir, is it possible that from the statistical data which we have and the phenotypical results which we have, can we combine them and make a software using mathematical models at the back end so that it can help in the early detection of the cancer?
So based on imaging data, yes, I think people are trying out similar approaches from what I'm aware of. There's something called digital pathology, which is becoming increasingly popular these days. There's a lot of machine learning and deep learning going on when you give pathology images to a computer and then sort of see if the computer can learn what patterns to associate with different stages of disease. So there are many efforts going on in that direction. Again, I'm not sure if they have reached the clinical stage yet.
Thank you, sir.
all right. So thanks, Mohit, for this engaging talk. And now that we know how can we relate science and memes, right? So others can try that too and experiment and see what is the outcome. So for Funday Sunday, season 2 has started, and please do submit your entries for Funday Sunday. We'd love to have your submission and see what you have in mind for creativity in science. And do visit our page for Funday Sunday on India Bioscience website. That's it for today, and have a good evening. Bye-bye.
Thanks so much, Suchil.
Yeah, yes, Mohit.
Now just saying thanks so much, Suchil. Yeah, thank you.
Thank you. Thanks for joining. And thank you for joining, everyone. Today, we have Shantala and Jile from India Bioscience with me. And please do submit your feedback. Here's the link in the chat box. Thank you.